cylinder.
Yes, sir.
Okay. So it is a hollow cylinder. It's about 500 nanometer long. 500 nanometer long cylinder. It is like this,
Yes, sir.
like this.
Yes, sir.
All right. So each centriole has 9 triplets of microtubule. This is the one microtubule that we have seen on this screen. So there are about 3. It is a triplet
Yes, sir.
like this, like this, right? So as we know, there are 2 centrioles which are present in the cell, and they are present in a membrane of the structure that is called centrosome. Like this. So here you may have 1, 2, 3, like this. This is the one structure, and there would be another structure which would be on 90-degree angle that will be present here, and it is also having triplet structure of triplet cylinders or microtubules like this.
Yes, sir.
Like, so this whole structure in which these 2 triplets of microtubules are present, this is what we call centrosome.
Yes, sir.
Centrosome is actually, some is a body. Some, the term some that we commonly use for body. So it is
Yes.
a centrosome. Are you getting this point? Okay. So now let's move on the next point, that is its function. What is its function? What is its function? So what is its function, man? It helps in what? Theories.
सर जी स्पिंडल फाइबर्स बनाने में.
Cell division. Remember.
Yes, sir.
Why those spindle fibers were formed in the cell? The basic purpose of those spindle fibers was to divide that cell. Actually, meiosis or mitosis, spindle fibers are actually formed in meiosis or mitosis.
Yes, sir.
Are you getting? So, and this process is always in those cells which are going to divide, right? So in this case, its function is it helps in cell division. As we know, this centrosome in which there are 2, it's a pair of centrioles. As we know, in each cell, a pair of centrioles are present. So when this, for example, it is present in a cell, here you have a cell in which this centrosome is present, and when this cell is going to divide, so this centrosome will disintegrate, and these 2 uh, centrioles, they will migrate on the opposite poles. The one centriole will move on one side, the other will move on the other side, right? So
Yes.
from there it will form fibers on which the chromosomes will arrange, and it will further go to divide and replicate whatever it is. Okay,
Yes, sir.
so in this case, its function is to help in cell division. So whenever a cell is going to divide, so this centriole, this centrosome will disintegrate, and both of these cylinders, triplet cylinders, will move on opposite poles, and they will help in the formation of spindle fibers.
Yes, sir.
So we can also add another thing here. One thing is helps in cell division. Another point that you can add, that is forms spindle fibers.
Yes, sir.
Forms spindle fibers. Are you getting that?
Yes, sir.
Is it? And what is the function of these spindle fibers in case of cell? If you know about this, what do you think?
Sir, she
Hmm.
prevent the the वो क्या होता है जी,
Okay. Think about it.
सेंट्रोसोम्स को पुल करना साइड्स पे.
Chromosomes,
हाँ,
because
क्रोमोसोम.
chromosomes, the chromosomes which are arranged on the spindle fibers, uh, centrioles will pull the spindle fibers back to on the opposite poles. The purpose is very simple. The purpose is to divide the chromosome number which is present in the cell, right? So this is you must keep in mind. All right.
Yes, sir.
So what type of question they ask you in from here? It's related to function. Anyway, so next point is that, right?
Yes, sir.
This was all about centriole. You must know what is centrosome, what is its structure, and how it functions. This is what centriole is. Okay. So now let's move on the next point, that is cell wall.
Yes, sir.
Okay, cell wall. So what do you know about this cell wall? What's the previous knowledge about this cell wall? Do you have?
Sir जी,
What it is,
sir,
its specificity, where it is present. Yes, it is. Please, please.
sir, cell wall is present uh, out at the outermost. It is the outermost layer of the cell before the cell
Which
membrane.
cell?
And
Which cell?
its function is to provide sir, mechanical support or not mechanical support, yeah.
But where it is
Structural
present?
support. Sir, it is the
Is it
outermost.
outermost, but in what case? Whether it is present in all the cells, all types of cells, or it is specific to some cells?
In plant cell.
Yes. Not in animal cells or
Yes, sir.
only in plants.
Yeah.
Okay. So cell wall. Cell wall. Cell wall. Cell wall is about 2 micrometer thick.
Yes.
2 micrometer thick. All right. One thing. And the primary cell wall is rigid and made up of parallel polysaccharide cellulose fibers. We know that. Another thing is its composition.
Yes, sir.
Composition. So it is made up of cellulose. Talking about plants. While in case of other, like if we are talking about fungi or some other cell walls, like in bacteria, so they have different composition. But in case of plants, the composition of cell wall is cellulose.
Yes, sir.
All right.
Yes, sir.
So what is cellulose? Cellulose is actually a it is polysaccharide, right? So this structure is like there are some parallel fibers crisscross. This arrangement is crisscross arrangement, and these are the parallel fibers, like the fibers like here arranged like this. So it is a polysaccharide and form a specific structure that is called cellulose. And definitely its structure is very according to its function. So we will talk about all about that.
Yes, sir.
Cell wall is fully permeable, right?
Yes, sir.
And it has high tensile strength because it is crisscross structure. So it has very strong mechanical ability, very strong mechanical property.
Yes, sir.
Cell wall is fully permeable. What do you mean by this? It has no ability to stop any component to not to get it. It has not any to filter the substances which are going into the cell, right? That's why we call it, it is fully permeable. It is fully
Permeable.
permeable, right? And another important thing, that is it has high tensile strength. What is tensile strength? We have studied in physics. Ability of the substance break by applying force,
Yes, sir.
right? So when you will apply any force to break this cell wall, so it will take, or it will uh, consume a lot of force, or it will bear a lot of force before going to break, right? So it has high tensile strength because uh, it provides cell to keep itself intact, or it protects the cell from outside forces, from pressures, right? Because plant is always exposed, and every type of pressure that plant has to bear, and uh, the main role in that strength is provided by this cell wall. All
Yes.
right.
Yes.
So so the first one was these horizontal lines. These were actually primary,
Okay, sir.
and when you are crossing the fibers on it like this, these fibers,
Yes, sir.
they are forming secondary cell wall. So primary cell wall and the secondary cell. So this structure that you are observing right now on the screen, it is a complete cell wall which is made up of a primary and secondary structure, and it is a crisscross appearance. And this crisscross appearance is actually giving it strength in order to bear all the pressure from the outside. And even there are some cell walls on which another chemical may also present, that is lignin, right? This
Mm.
lignin is actually providing this cell wall more strength. And where it is not present in all types of cells, but it is present here in xylem.
Okay,
As
sir.
you know, xylem has to bear a lot of pressure of water,
Yes, sir.
right? So
Yes.
this water pressure, it's this water pressure on the xylem uh, make it, you can say, make it more capable to have a lignin for further strength. I mean, this xylem is strengthened because of this additional chemical, that is lignin, because it has to bear a lot of pressure of water. So this water actually compel this structure to be more modified or to be more strengthened by having lignin in it. Okay, so all other structures do not have the lignin. All right.
Yes, sir.
So this was the cell wall. Again, you can, I'm concluding this composition. I have told you there are uh, two layers of cell wall. The one is the primary cell wall, which is parallel structures of the fibers and which are arranged horizontally. And the second layer is the secondary cell wall, which is arranged on, you can say, in a longitudinal way, uh, having the fibers which are already arranged here parallel, in a parallel manner, and this is how it forms a cell wall structure. And the structure cell wall of the xylem, that has additional chemical, that is lignin, that provides more strength to the cell wall. So now we can talk about its function. What's its function? जी बेटा, what is, what's its function?
सेल वॉल का सर जी यही फंक्शन है कि प्रोवाइड्स स्ट्रेंथ और मेरे ख्याल में स्टोमेटल ओपनिंग में ये हेल्प करती है.
Strength, okay, strength is fine. How it helps in stomatal opening?
सर ये वो गार्ड सेल्स में इसके जो डायमीटर होता है वो चेंज होता रहता है, जिसकी वजह से एक सेल जो है वो फ्लेसिड होता है या फिर वो एक्सपैंड करता है.
Okay, all right. The first thing is very primary role of this cell wall is actually to provide mechanical strength.
यस सर.
Mechanical strength. It is primary function, and it is a very primary and important function uh, of the cell wall, right? Because of this mechanical strength, it remains or it maintains its turgidity,
यस सर.
right?
यस
Yes,
सर.
turgidity.
सर जी टर्जिडिटी होती है कि उसको ब्रस्ट होने से बचाता है.
Of the cell because of water, internal pressure of the cell, which is pressure which is you can say which is towards the wall of the cell wall, right?
सर.
For example, you have a cell here and you have water molecules inside, so these water molecules put pressure outward in the cell to in order to maintain its shape, so that it can maintain its shape. Why this shape is maintained? Because these water molecules inside, they are putting force, or you can say putting pressure on the, and the cell wall is also present outside, is also providing its turgidity. So in this case, turgidity is the most important element of the plant. Another important thing, that is it prevents bursting,
यस सर.
prevents from,
ब्रस्टिंग.
it is not brushed because there is a very strong layer which is present on the cell, that is cell wall, right? Another important factor that is providing that is the part of this, that is it helps in transport. How? uh, they provide some route. We will discuss this in the chapter of transport, but here we just need to know there is a route, or there is a pathway that is called
प्लाज्मा.
apoplast.
यस सर.
Apoplast. This pathway of transporting of water from one cell to another, this is through cell wall.
यस सर.
We will discuss its detail, but right now we just need to know there is a pathway or the route through which the water is moving from one cell to another, that is the apoplast, and it is through the cell wall. And another thing, plasmodesmata. Plasmodesmata. What is this? Plasmodesmata is actually connection between the cell wall. Connection between the cell walls of the neighboring cells, of the neighboring
सेल्स.
cells. For example, you have a cell here, so there will be a connection between these two cells through this cell wall. This is what we call, for example, here is a cell, here is another cell. So there is a connection between these two cells, that is plasmodesmat. This is plasmodesmat. right? So this interlink between these two cells, this is called plasmodesmata, and this plasmodesmata is actually cell wall. It is the extension of cell wall, remember. It is the extension of cell wall between these two cells, right? So it is a function of cell wall.
Yes.
Okay, so you can add something else as well here, like it is non-lignified connections in plants, and they transfer the material from one cell to another. Definitely, I have told you that its main function is to transfer material. For example, there is a molecule on this side, some water on this side. So if it has to move from this cell to this cell, so this is a connection actually, and this connection will help these molecules to move from one cell to another cell. So
Yes.
without this plasmodesmata, these molecules, these two cells cannot communicate with each other, or they will not transport any substance from one place to another place, or from one cell to another cell. So
Yes.
this was the function of the plasmodesmata. Or the whole, we can summarize the function of the cell wall. The first function is it provides mechanical strength,
Strength.
it prevents cell from bursting,
Yes, sir.
and it helps the cells uh, to you can say
Drainage.
have a route that is apoplast.
Yes, sir.
Apoplast is the route which is major transport route, right? It is a transport route actually.
Yes, sir.
Transport, right? So
Yes, sir.
any type of transportation will occur through this apoplast, and this is because of this cell wall. Another important things I have told you, that is of its connection of this matter. This is the connection of between these two cells, and this is through the plasma this matter. All
Yes.
right.
Yes.
So for example, you have a cell, so definitely there will be a nucleus in these two cells, and there is will be a cytoplasm and other things. So this is how it's going. So another point that we need to know is vacuum. What is vacuum? Anyway, uh, so always just try to recall the knowledge that you have about the vacuum. What it is? What do you think when you listen this term or to hear this term? So, what the first thing that you get in your mind?
Vacuole surgery.
That yes, what is
Surgery,
vacuum?
the innermost part of a plant cell.
Okay, it is the part of the plant cell, definitely.
Uh,
So, what do you think? What type of what type of shape that you can assume uh, of this vacuole? Like it is a balloon-like shape structure, uh, membranous structure
sir, it is a
present. Hmm.
uh, membrane bond structure, and it's, I think, present in animal cells and plant cell both. And
Both in, yes, it is present in both cells,
yeah,
but there is a difference that we need to understand that difference as well.
yes, sir.
And vacuole is actually, as you told now, uh, vacuole is a membranous structure uh, like uh, like this. It may be, if you are talking about plant cell, animal cell, if you are talking about the plant cell, so there is a difference. Here there will be a number of vacuoles in small size, but in case of plant, uh, vacuole is a large structure. That's why uh, in normal cell, the nucleus is present in the center of the cell, right? But in case of plant, nucleus is present. This is on periphery because of this large vacuole, because the space is occupied vacuole because the nucleus is slipped little bit towards the periphery of the cell. Are you getting this point?
Yes, sir.
So animal cell have large number of small vacuoles. Lives phagocytic vacuole in phagocytes, food vacuole, lysosomes. So, for example, there is a cell. Its function is to phagocyte. Do you know what is phagocyte?
सर phagocytes वो जो engulf करते हैं.
Yes, engulfing ability of the cell. So it means these vacuoles are, for example, the cell in which this phagocytic, uh, we call it phagocytic vacuole, right? It is present in a cell in which this has the ability to engulf the substances. So phagocytic vacuole in phagocytic, food vacuole, lysosomes, like it is also present in lysosomes in the cells in which the that has the capacity or that has the function to phagocyte. While in case of plant, it has the large vacuole, and its function is actually most of the time its function is storage. We will talk about this function as well, other functions as well. But right now, we just want to know there is a bag or balloon-like structure. Its size may be changed. Its size in case of plant is large, while in case of animals it is small. In case of animal, there are more than one vacuoles. Number of small vacuoles are present. While in case of plant, there is only one vacuole, and this vacuole uh, is a larger in size, and it has occupied the major portion of the cell of the plant. Right?
Yes, sir.
Are you getting? What is its function?
Vacuole
Yes.
in an animal cell.
Provide
Uh, s-
provide turgor pressure when
okay.
water enters by osmosis.
Yes.
This cell will be a turgid when definitely water is coming from outside. So when this water is coming from outside, this water will get into the cell, and if the water is extra in the cell, so what cell will not utilize it, but push it towards the vacuole. A vacuole will be filled by this water through the process of osmosis. Osmosis. Osmosis is the process in which the water is moved from higher concentration to lower concentration through the semi-permeable membrane. So this membrane will get in this water into the vacuole, and this vacuole, which was in a small size, but when this get water, so it will enlarge,
Yes, sir.
right? So when it will enlarge, it will automatically enlarge the cell or make it turgid, right?
Yes, sir.
It also contains hydrolytic enzymes act as a lysosome.
Okay, sir.
It contains hydrolytic enzymes. And where were these hydrolytic enzymes were present? In the lysosomes. And lysosomes were also like these vacuoles. So they were also one single membranous structure in which the hydrolytic enzymes were present. So these vacuoles may also act as a lysosome
Yes.
if they have the hydrolytic enzymes. They may also contain some various chemicals that may accumulate inside of this vacuole, because there are certain, like it is possible that it may contain some you can say some metabolites, which are the chemicals, the substances which are produced by the process of metabolism. They will be stored in the vacuole, like latex, like latex. It is latex. Okay, it is accumulates in the vacuoles. Or there are many other alkaloids. They are also stored in the in this vacuum. Okay, fine. So another thing, that is different pigments. Pigments. Where should I? The color that you see
Yes, sir.
in flower and fruits,
Yes, sir.
right? Those colors are mostly in the form of anthocyanins. This is a pigment. This anthocyanin is present in the vacuole of the plants, which actually give different colors to the fruit and flowers, right? So it is also present in the plants. So this vacuole is actually you should need to know about its function. That function, its major function is of storage. If you see all these functions, the hydrolytic enzymes, it contains metabolites, it contains pigments. So all these are actually stored in the vacuole of the plant cell, because that is the large cell. So it has the ability to extra material which is not essentially or which is not immediately utilized in cell is commonly stored in the vacuole, and it is released when it is required, right? So for example, it stored water inside. So when water is required, when there is a deficiency of water, so this vacuole will release water, and as a result it will uh, compensate the requirement in the cell. So this is how this vacuole is performing its function. Do we know about the eukaryotes and prokaryotes? I think we have already
Yes,
discussed
sir.
about this.
Yes, sir.
It's the size of the you can say size, nuclear membrane. You know about this, what type of structures are present there, cellular circular DNA. We have, I think, discussed about this. Do we need to talk about more?
No, sir.
What are
No.
so cell wall, the difference of cell wall you must keep in mind. The
Yes.
cell wall, which is made up of murine in case of prokaryotes.
Yes, sir.
Let me write something. Okay, I'm writing murine. Right.
Yes.
So it's another name. For example, uh, in case of gyropeptide, so here urine or peptidoglycan, by which this structure is formed, right? In case of prokaryotes, but if you are talking about the eukaryotes, okay, in case of eukaryotes, you would have uh, the cell wall which is made up of cellulose. Okay, Baki 70s, 80s difference. So I think in case of prokaryotes, you should know about the difference. So I think the one thing that we will discuss in the tomorrow's lecture, that is the structure of virus and how protein is synthesized. And I think you know very well how protein is synthesized, but we will just give a short glimpse to this. And at the end of this, we will discuss a worksheet, right? So this chapter will be concluded, hopefully. All right.
Yes, sir.
Okay. Please, anything you want to ask me?
No, sir.
ये थोड़ा सा short चीजें थी, but remember, don't ignore them,
Yes, sir.
because these are the very basic concepts in biology. Because if you know, if you don't know about the structure and function of the cell of different subcellular structures, so then uh, I think you can't build the concepts regarding which are more complex or related to the cell. So at least you should know about the structure and its few functions, right?
Yes,
So
sir.
so that it would be easy for you to go on the further or the more complex topics. All right,
Yes. Yes. Okay, sir.
fine. Anything else?
No, sir.
Hmm. So प्रीवियसली तो मैं जो ऑनलाइन बच्चा था, बच्चे होते हैं हमारे पास नॉर्मली, हम ये भी करते हैं कि हम उनके साथ एक टेस्ट भी कंडक्ट कर लेते होते हैं हम वीक पे। मतलब अभी मैं इसलिए आपके, चूंकि आपने ये किया हुआ है कि दो सब्जेक्ट्स को एक ही उसके अंदर डील कर रहे हैं, सो आई डोंट नो बट आई एम ट्राइंग माय बेस्ट टू डिलीवर यू मैक्सिमम।
Yes, sir.
और ये है कि हम फिर थोड़ा सा टाइम लेके फिर हम आगे टेस्ट वगैरह भी कुछ ना कुछ वीक में एक करेंगे, जो कि वर्कशीट्स पे बेस्ड ही होगा, वो दैट विल बी योर प्रैक्टिस।
Yes, sir. Okay, sir.
ओके, अरे सी यू,
Okay,
सी
sir.
यू टुमारो, चलो, ओके।
Yes. Okay.